Thank you for the invitation. And once again, thanks to all the organizers. The topic of my talk is a large European survey on unsuitable samples that our working group for pre-analytical phase has done some time ago. I will be sharing with you results of that have been published in two subsequent papers, so part 1 and part 2. The part 1 addresses the way that laboratories in Europe monitor pre-analytical phase and also how these data from this monitoring are currently used. And then part 2 addresses in particular the way that laboratories detect and manage hemolysis, lipemia, and icterus. This has been a large survey, and we altogether have collected responses from 1,347 laboratories from 37 European countries. Out of those laboratories, most of them were hospital laboratories or laboratories that serve both primary care and hospital, and only a minor proportion of the laboratories were primary care laboratory, and most of them were public, non-profit laboratories, and only 1/3 were privately owned laboratories. Most of those laboratories were so almost, I don't know, 80% were either small or medium-sized laboratories, and only a minority were laboratories that receive a couple of thousand of samples per day. Most laboratories had some kind of accreditation according to ISO or some national standards, and only 1/5, so 20% of laboratories did not have any accreditation or certification. We also had 18 laboratories from Ireland, which is 1 point of all laboratories in the study. Out of those laboratories that we surveyed, 94%, which we felt was very nice, were monitoring pre-analytical errors. And Ireland is here, and it seems that 100% of laboratories that participated in the survey are monitoring pre-analytical errors. Out of those laboratories that do not monitoring, so those 6% of laboratories that do not monitor pre-analytical errors, we asked those laboratories, what are the reasons for not monitoring pre-analytical errors. And I'm trying to hide this, sorry, but it doesn't go away. Anyway, out of those laboratories which did not monitor pre-analytical errors, there were laboratories that did monitor those errors because of the human resources, or because of some technical issues, or because they felt that there is no need to monitor those errors, etc. What was interesting to learn is that nearly half of those laboratories who were not monitoring those pre-analytical errors declared to be accredited or certified, and we felt that this is really very strange. Sorry. Now, labs that monitor pre-analytical errors were further asked, okay, you monitor pre-analytical errors, but do you perform further evaluation and statistical analysis of the frequency and trend of those data? And, it was okay to learn that large proportion, so almost half of the laboratories, do some kind of periodic evaluation, monthly, quarterly. However, we were very surprised, unpleasantly surprised to learn that 1 out of 3 laboratories, so 31% of laboratories, don't do any further evaluation of the pre-analytical errors. And 23% did some kind of irregular evaluation when, there is a demand, so not on a regular basis. And we felt, this is really strange. When we asked laboratories that perform some further evaluation and statistical data analysis, so 875 laboratories which do periodic and irregular evaluation, so those 2/3 of laboratories, when we asked them, do you have a process for follow-up and taking actions if those pre-analytical values worsen or are outside of your defined cutoffs, then again, it was very surprising to see that 1/3, which is, you know, more than 30%, didn't have any process for follow-up and taking actions of those errors. So they do monitor, they do even periodic evaluation or sometimes irregular evaluation, but then even when they analyze these trends, they don't have a plan and they don't do anything about those errors. What was really very surprising for us was the fact that even some labs that were accredited and or certified were not really dealing with those pre-analytical errors properly. So out of the labs that were accredited according to ISO 15189, you know, almost 2% of those laboratories didn't do any monitoring or documentation of pre-analytical errors. 26% didn't do any statistical evaluation of those errors. 40% didn't have any process for follow-up. And 16% didn't take any action if those errors were worsened or you know, would fall outside of some defined cutoff. So we felt that these numbers are really surprising, and the same was with the 17025 or with ISO 1901. 9001. So, you know, the conclusion was that probably the auditors that were assessing those laboratories didn't look into these, into the pre-analytical phase, and didn't have same standards and same criteria when assessing laboratories in comparison to some other assessors in Ireland, but also outside Ireland and in Europe, across Europe. So we felt that there is something that needs to be done regarding that issue, and I will get back to that problem later during my presentation. The part 2 of the survey dealt with the questions that relate to the way that laboratories manage hemolysis, icterus, and lipemia. So we asked, you know, do you monitor HIL indices? And it was in a way good to know that majority of laboratories are monitoring HIL indices. So 86%, and you could say, well, this is fine. You know, quite a lot of laboratories do monitor HIL indices. However, you know, if you think the other way around, there are still quite a number of laboratories, 14% of laboratories that do not monitor HIL indices, and we felt this is too high. Well, when we asked those laboratories that do monitor, so these 86%, do you monitor hemolysis, icterus, and lipemia? When we ask them, okay, how do you measure the degree of hemolysis, lipemia, and icterus in your laboratory? Then again, not something that we felt was quite disappointing was the fact that 1/3 of laboratories actually were still measuring the degree of hemolysis by visual detection, and only 40% were using automated HIL indices, and 1/3 used a combination of the two. When we asked those laboratories that used visual detection, you know, do you use a color scale for this to assess the degree of hemolysis, or do you just assess the degree of hemolysis by your own subjective criteria? Then again, you know, we felt this was not really acceptable. 2/3 of laboratories didn't use any kind of color scale. So they were just actually assessing the degree of hemolysis very, very subjectively. And only 1/3 of laboratories did use some kind of color scale to standardize the way that the degree of hemolysis was assessed. And this is wrong and problematic because we know there are a number of reports showing that visual assessment is highly unreliable, and that the visual assessment poses a risk to either reject acceptable samples, so samples that should have been analyzed, or risk of accepting samples which are otherwise not suitable for analysis. And this jeopardizes the patient safety. So we felt this was quite disappointing. When we ask now, a further problem is that we even don't agree about the definition of hemolysis. So those laboratories that monitor the frequency of hemolysis, for example, and frequency of other preanalytical errors, do not agree on how do we define hemolysis. So what do we consider a hemolyzed sample, and what do we count as a hemolyzed sample in our statistics? And there were the answers ranging from, hemolyzed sample is anything from 0.1 grams per liter to 0.3 grams per liter to all the way to 1 gram per liter. So there is a huge difference in what we define as a hemolyzed sample. Moreover, we also manage unsuitable samples, so not only hemolyzed, but also lipemic and nectaric samples very differently if we look at the all labs that responded to the survey. So, there are, if you look hemolyzed samples and the way we manage hemolyzed samples, then there is a minor proportion of the laboratories, somewhat less than 10%, which would reject the whole sample if the sample is hemolyzed. Then there is, on this right side, there is a minor proportion, a couple of percent of laboratories, which would release all tests. So they wouldn't report even that the sample was hemolyzed. And even though this is just a couple of percent, so maybe 2-3%, we are talking about 1,000 samples. So 2% of 1,000 samples is, I don't know, 20-30 sample laboratories. So this is not really something that can be neglected. And then there is, again, a huge number of laboratories that would release all tests, even though they would be affected by hemolysis, and they would just add a general information of hemolysis. And then there is, luckily, the largest proportion of laboratories which would release only, reject only affected tests and perform all other tests in this hemolyzed sample. So, but there is a huge difference in the way we do that. We manage those unsuitable samples. Not only there is a huge difference in the way we do it, but we also have very different criteria or very different cutoffs to decide when to do what. So to manage unsuitable samples, to decide, whether you will accept or you will not accept a certain sample, a particular sample for analysis. So while more than half of the laboratories would use parameter-specific cutoffs provided by the manufacturer, there are a large proportion of laboratories that would have something else, like, for example, in-house developed cutoffs, or one fits for all, so same cutoff for all analytes. Or there is a combination of there are a certain number of laboratories which would do the HL detection, but then after that they would do a visual check and then make an individual decision, which is highly unstandardized and subjective. And then there is 1 in 5 laboratories would do individual decision only based on visual check. So there is a huge difference in the way we do that, and this can have substantial consequences to the patient results. When we asked, so those, you could say, well, it's good that more than half of the laboratories that responded to the survey were using parameter-specific cutoffs provided by the manufacturer. And you could even say, well, this is the way we should be doing it. Well, in a way, yes. However, what we should know is that those cutoffs should be verified. And when we asked those laboratories, did you verify those cutoffs declared by the manufacturer? Then 60% of laboratories didn't verify those cutoffs. So they would just use it without any verification. 17% would verify them, and 22%, 17 would verify all of them, and 22% just some of them. But major proportion of laboratories that responded to this survey did not verify those declarations. So they would use it without any reservation. And why is that a problem? Because there are many reports in the literature that have demonstrated that these manufacturer declarations cannot be verified in the lab. So if you take, for example, declarations for hemolysis, which was the aim of this group from Spain. And if you try to verify on different instruments, then you will see that there are a number of declarations that cannot be verified, like all those in red. They were not verified by the laboratory on the same equipment. And we did, our group in Croatia did the same or similar project with the colleagues from Italy on declarations for lipemia. And we also learned that if we used 3 different instruments, and we were not able to verify all declarations by the manufacturer. So those declarations are not quite appropriate to be used without a prior verification in the laboratory. So what we learned from that survey is that management of unsuitable samples in Europe is not quite satisfactory, and that there is a lot of room for improvement. And we felt that the way forward would be to harmonize pre-analytical practices, to improve the way that pre-analytical quality is managed, and also to harmonize criteria for quality audits, because as I told you in the beginning, there were a number of laboratories that were accredited, which didn't really monitor and deal with pre-analytical errors as the standards actually requires. So the EFLM working group for pre-analytical phase has done some work to assist laboratories to better manage pre-analytical quality, and to, in particular, the management of unsuitable samples. So we have published a couple of papers, and I will just briefly mention those papers because we don't have time to go into details. So the first paper is relates to provides actually recommendations for how to manage hemolyzed samples in clinical chemistry testing. And this paper provides recommendation on how to systematically assess serum indices, how to define H index cutoffs, how to decide when to flag and how to flag reports, and when and how to alarm, when and how to suppress hemolysis sensitive tests, etc. The other publication that we published that relates to the problem that I just highlighted is a paper that provides an expert opinion about the way that we as a working group believe that internal quality control for HIL indices should be managed. So here we provide advice on how should you implement and how should you use internal quality control for to monitor your HIL indices. We also addressed the issue that I just mentioned that relates to manufacturer declarations. And with this paper, we call for more transparency in manufacturers' declarations on serum indices. And we also aim to propose, so we bring in this paper some solutions that we recommend should be adopted by manufacturers to increase worldwide harmonization of serum indices. And last but not the least, this is our most recent paper published just a couple of months ago. With this paper, we are addressing the ISO auditors, but also laboratories. And with this paper, we aim to provide an overview of the ISO 15189 pre-analytical requirements, and also to provide guidance for laboratories on how do we believe should these requirements be met. And in this laboratory, you will, in this paper, if you go and read the paper, you will see that we have provided a huge table with we have listed all pre-analytical requirements, and we have provided the minimum, what we believe is a minimum solution, and also the best-in-class solution for each and every pre-analytical requirement. So to conclude, unfortunately, the results of our survey show that we are dealing with unsuitable samples in Europe quite differently. There is a lot of heterogeneity and a lot of bad practices, and these bad practices affect patient safety. So we obviously need to do something about it. We need to improve. And our working group has done some work to help, and we'll do some more. So keep calm, and the new stuff is going to come very soon. Thank you very much.
Hi, That was fantastic, Anna Maria. There's a lot of questions coming in, so I'm hoping you can stay with us till 12 o'clock so I can start throwing some awful questions at you, if that's okay. Just, I was going to ask you one thing, if you don't mind. This is just out of sheer nosiness. When we have a lot of inspections in Ireland, we don't get asked so much about the pre-analytical phase. I'm just wondering, in inspections in Croatia, would it be the same? Would they focus very highly on it, or would they kind of shy away from it?
Well, I think the focus is increasing, and this survey actually shows, because as you could have seen, a lot of pre-analytical issues were not really managed as they should be, and all those laboratories were accredited. So obviously no one didn't ask, are you doing that? So I think it's increasing, and I like to believe that our working group helps in raising awareness about the importance also of the pre-analytical phase. And with this paper, we also tried to raise awareness also in auditors, not only in laboratories, but in auditors. These are the things that you should be looking at, and these are the solutions that should be there as a minimum. So we believe that, there is going to be some improvement in the future.
Excellent. And if you're ever looking for some Irish representation, just come to PALMSoc. We're all game. Thank you very much. So will you hang on?